Great. Thank you very much, and I'd like to thank the organizers for the opportunity to speak this afternoon. Afternoon, everybody. I know it's been a long afternoon of talks, but thanks for bearing with me. I want to talk this afternoon about Quell Therapeutics, which we are a clinical stage engineered Treg company. And so I'm going to switch gears and talk about T regulatory cells, which we really haven't spoken about at this conference yet, and really how we're using them in our transplant and in our different products in our pipeline. So really, I just want to take the opportunity, because I think I'm probably the first one that's spoken about Tregs here. Really, why Tregs? So unlike T effector cells, which actually can be pro-inflammatory and actually act to kill tumor cells, and we're all very familiar with CAR Ts, Tregs are really modulators of the immune system. So they do the exact opposite. So they really control immune responses, teach other immune cells how to play nice and how to behave well with each other. And they really do this through multiple mechanisms of suppressive activity, both direct and by bystander in the local environment. So they really have an effect on antigen-presenting cells, other T cells that come into the area. So you're really getting a pharmaceutic response from these cells that you can't alone get, with a biologic or with a small molecule. So we really think, because of this, using Tregs, we really have the ability to durably reset the immune system with a one-and-done approach to delivery of the cells. And I'll talk about that when I get to our liver transplant trial. And just a bit about Quell, for those of you that don't know, we're a London-based biotech company. I myself am based in Boston, but the majority of the company is in London. And our first trial was approved by MHRA, Belgium, and Spain, and we're currently enrolling and treating those patients right now in the UK. We have our own GMP manufacturing capacity in London, and we have the opportunity to then demonstrate, hopefully, if this trial that can be tolerogenic in liver transplant patients, and I'll talk about the unmet need there and really getting these patients off their immunosuppressive drugs. We have about 140 employees in West London, and to date, we've generated about $300 million in capital of investments, and that really will take us through to our clinical milestone at the end of next year. So why Tregs? I spoke about this a little, but just to give you a little bit more flavor of what we're talking about, as you'll see in the center of this diagram in the orange, and this will become important in a moment, is really FOXP3. And what is FOXP3? FOXP3 is the master transcription factor which regulates the function of Tregs. So Tregs have this that come on in terms of development, and it's important actually when we ex vivo engineer these cells that we make sure FOXP3 stays on. So we actually have in our construct extra copies of the FOXP3 to make sure that we lock these cells in what we call as a phenotype lock. And it's very important because FOXP3 plays into all of the functions that you see in this diagram. So the ability of the cell to maintain this and maintain its suppressive function, also have an effect on dendritic cells and convert effector cells to suppressor cells, really is evident in all of these areas that it can do. And over on the right is an assay that we use a lot for our release and our testing to make sure that the cells that we're getting from the patient and that we expand have this suppressive ability. And it's, you look at the percent suppressive in the white box on the y-axis, and then we mix then our cells in diluted ratios across the x-axis in what's called a mixed lymphocyte reaction, and we're measuring then the ability of the Treg to stop the proliferation of effector cell, to suppress that function. And that's what you're looking at. So we want to make sure that our cells are able to, after expansion, suppress in vitro, and then also we use these in several modalities. Transplant is the first that I'll talk about, but also in the immune space, and then our next-gen program in terms of allo. And what you're looking at there is just our lead product, an animal model in which we tested this, which is an A2 CAR, which takes advantage of the HLA mismatch between the donor graft and, in this case, an animal, but in our trial that I'll talk about in the patient. And you can see by delivering A2 CAR Tregs, you are able to make that A2 positive graft survive in the absence of immunosuppression. So this was the hallmark preclinical data that led up to our trial. And really, the opportunity in transplant, there's a huge unmet need. Even though you think that patients are well served, and maybe before I joined Quell 2.5 years ago, I thought transplant patients were well served with immunosuppressive drugs, but it's really what we want to get them off. Because believe it or not, liver transplant patients, and as I'll talk about in a moment, have the same comorbidities and the same mortality as most of the common malignancies. And really from the morbidity that's associated with the heavy immunosuppression that these patients are under over the lifetime of their graft. So really, Quell 001, which is our first product, which is an autologous engineered A2 CAR Treg, and our first study, our phase 1, phase 2 study, Liberate, is what the goal is, to try to liberate these patients from all immunosuppression. And then we move on to autoimmunity and inflammatory conditions. And I think everyone in this audience realizes what a large population autoimmune disease is, and the market, and the opportunity really to move beyond biologics, which are treating the symptoms and not really be disease modifying. So we announced in the summer that we entered into a partnership with AstraZeneca in order to develop these same engineered Tregs in the area of type 1 diabetes and IBD. And I'll talk a little bit about that at the end. But just so that you understand our platform, as I said, it's an autologous engineered Treg platform. We take Tregs actually out of the patients from leukapheresis, very similar to CAR T development. And we have our own GMP manufacturing, as I said, in London. Very critical to our multi-modular platform, in which we put these genes under an exogenous promoter, driving off of one construct, and lentiviral transduce it into the cell, is we have different modules. The one in orange is super important, I'll talk about in a moment, which is the FOXP3 phenotype lock, which I already introduced, and that maintains the function of the Treg. Antigen specificity we can do with a CAR or a TCR, and I'll talk about our CAR programs. And then depending on the disease, we have different modules that we can plug and play. If we need persistence, we put in domains of IL-2 receptor that drives STAT5 signaling. If we think the cells need to see IL-2 in areas of the brain and others where IL-2 may be limiting. Also armoring techniques, and our product has a safety switch. So the lead product has the phenotype lock, A2 for the antigen specificity for CAR, and a safety switch. And one of the very early on decisions that we had to make as a company is what is the source of the Tregs that we're going to use? And even though Tregs are, a paucity, in the blood, we still feel that it's really important to start with a bona fide naive Treg. And the reason is at this data here. So what you're looking at is we did transcriptional analysis, and we took T effector cells and we measured what's going on in their transcriptome by PCA analysis that you're looking at here, and said, okay, what does a normal T effector, their transcriptome look like in green? What does that happen if we put a CAR on that and overlaps that in yellow? What if we take our FOXP3 cells then that are T effectors, put a CAR on them and overexpress FOX, which some people are doing because effectors are more abundant in the blood? You can see it moves to the left a little bit to match the transcriptome all the way over in the left of a Treg in purple, but there's still massive daylight between those populations. And if we CAR our bona fide Tregs or overexpress FOXP3 in our bona fide Tregs, you'll see pretty much a Venn diagram in which they overlap. So really what I'm saying here is that we think we really need the phenotype following the function of the transcriptome, and by locking in and overexpressing FOXP3 in a bona fide Treg, you still keep a Treg, but you're not able to convert a T effector to a Treg just by overexpressing FOX. And really, I'll go through this fairly quickly because I want to get to the clinical data, and really the importance of overexpressing FOX in our phenotype lock. If you look at the bar graphs on the left, on the bottom are genes that are important for the function of Tregs. FOXP3 we already talked about. Helios is another one that's important for the stability. CD62L expression is important for the homing of the Tregs to inflamed tissues, and CTLA-4 is important for the immunosuppressive function. What you can see is, as I said, Tregs are very plastic. If you subject them to stress, and in this case, this is just cryogenic freeze-thaw stress during our manufacturing, the ones that are locked in the orange maintain all of the expression of these markers, whereas the ones in blue during the freeze-thaw lose these markers. And what happens when you lose these markers is you quickly lose expression and suppressive ability of these cells. And that's over on the right in the blue. The unlocked cells lose the ability to suppress in an MLR really quickly, and that is directly due to loss of expression and methylation of FOXP3, which you can see in the bar graph. And again, we asked the question, what happens to control cells? As with any cell therapy, we sort down to a very fine population, as pure as we want to get to the sorting of Tregs, but there's always residual cells that get carried over. So we asked the question, what happens to some of those contaminants in our manufacturing product? And in this case, our impurities actually are T effector cells, CD4 and CD8. So we put our phenotype lock on CD4 and CD8 cells that we isolated out of our manufacturing process. Then we stimulated, and then we asked, are they still able to behave like T effector cells and secrete pro-inflammatory cytokines? And you can see here that the ones that have the phenotype lock in orange, really the secretion of pro-inflammatory cytokines is ameliorated, whereas the untransduced ones still can. So it's an added layer of safety in our manufacturing protocol that we put this lock in. And then you can also see on the right that it additionally enhances the function of Tregs, in which you see in orange a better suppressive ability of these cells in a physiological range. The manufacturing, I'll just briefly go over. Most of you are probably familiar with manufacturing for CAR T. We start from the patient with a leukapheresis material. The one difference, as I point out, is the cell selection in the beginning. So we select down to only probably about 1 million cells. So we're only really transducing very pure, naive Tregs with our construct. We put in the transgene, which contains those 3 modules, including the CAR, expand this up in about 2 weeks. And we've now been able to do more than 30 GMP batches, both from normal autoimmune patients, transplant patients, and we're in the clinic right now with our safety cohort. So I want to talk to you about liver and why liver. I introduced this to you in the beginning, and really between the US and the 5 major territories in Europe, there's about 15,000 liver transplant patients per year. And those patients, as I said, are on lifelong immunosuppressive therapy. And that's what the graph looks like for the 10-year outcome survival of liver transplant patients. It's rather daunting. It's very similar to malignancies. They have infections, cardiovascular issues, and the nephrotoxicity associated with CNI. Really, these patients end up on dialysis and losing kidneys. So really what we want to do is really get the patients off all of their immunosuppression. There was a pilot study that was done in Japan, with polyclonal expanded Tregs against donor material. So they grew the cells alongside donor material. You can do this in Japan because it was a living donor. And that created a certain percentage of antigen-specific Tregs. But we're taking this a step further and engineering them specifically with a CAR. So as I said, our lead program has 3 engineered genes in them: the HLA-A2 CAR, the phenotype lock, and a safety switch, which is responsive to rituximab. And over on the right, you'll see how the trial design of our Liberate trial looks like. So again, it's important we're taking advantage of the A2 mismatch. So these patients actually be HLA-A2 negative and have received an A2 positive liver graft. That's about 25% of all of the liver transplants in US and those 5 major territories in EU. These patients are stable. They're 1 to 5 years post their transplant. We standardize their immunosuppression regimen. We apherese them then, get their cells. We'll give them low intensity conditioning, just mostly, it's very different. It's not to get rid of and make space, but it's really just to get rid of alloreactivity because these patients have had a transplant. Then we'll withdraw and change from TAC. We'll give the Quell 001 infusion. And over the course of several months, we will wean then off TAC. And at that point, at 2 months, post-immunosuppression withdrawal, complete immunosuppression withdrawal, we'll take a biopsy and we'll look at their liver, and we'll know whether or not these patients have operational tolerance, which they're able to maintain their graft. And then the endpoint of the trial is then 1 year off immunosuppressive therapy. But the important thing is, within the 2 months, based on the other weaning studies, most of the patients will have rejected during the wean or by this 2 months. So we'll have a very good read of what's happening in these patients. So this is a cohort of about 15 to 18 patients in which we'll be able to identify the cells. Importantly, we can biopsy the liver at serial time points here, as designated by the red bees. So we'll actually be able to go in, detect our cells by virtue of the CAR, by PCR, look for them in blood by virtue of the CAR or PCR. We'll be able to really understand the biology. Are the cells there? Do they maintain their phenotype? How long do they persist? And what are they doing in the patient for operational tolerance? So we have a whole set of biomarkers that are built in for this that I don't have time to go into, that will really allow us, for the first time with an engineered CAR Treg, I guess they shut off if I can't go back, sorry. I just wanted to show the pipeline, but I guess I don't have time. Is that we're also in autoimmune in type 1 and IBD, and I want to give a shout out to our collaborators, Solistic, in which we have an allo iPSC program in which we're generating an iPSC-derived Treg to use for future larger autoimmune indications, and be happy to chat with any of you at the break. So thank you very much.